Good day, Homo sapiens. How are you doing? Welcome back to studying Prescott's microbiology once again. So, today's topic is the bacterial cell membranes and the archaeal cell membranes. The cell membrane, the general cell membrane structure we have studied last time, and the general structure looked something like this. But today we are going to study about what is it, why the bacterial plasma membrane and the archaeal membrane are different from the general cell membrane structure. This cell membrane structure is, like similar to eukaryotic cell membrane, as well. Cell membrane doesn't have that much differences in them, right? So the general cell membrane structure had the peripheral proteins, phospholipids, the general lipid bilayer, and some kind of glycolipids or oligosaccharides, and things like that, right? We discussed the general structure of what the phospholipid actually is, what is its polar side and the non-polar side, right? Today we are going to study what is the difference from that structure, what is different in bacterial membrane from this structure, and what is different in the archaeal membrane from this structure, and how the bacterial and archaeal membranes are different from one another. So, to be honest, there isn't that much difference, but the few differences that are there matters, because it definitely points towards the activity of the plasma membrane of these microorganisms. By the way, if you don't know what the bacteria and archaea are, both are prokaryotes, both don't have true nucleus and the complex organelles, right? But bacteria and archaea are different, quite different, because of their ribosomal RNA. This distinct feature is make us clear that the bacteria and archaea are not the same things, even though in the previous time they were considered the same thing. And the other one is that the archaea usually live in the extreme conditions. It's because of their cell membrane structure. Today we are going to discuss this. So without further ado, let me demonstrate the few points here and there about those two, and then I'll read these or show you the diagrams, right? Now the bacterial membrane. The membrane structure that we studied here, the bacterial membrane differ because of the lipids that are present in them. You see these red components, these are known as hopanoids. These are the lipid molecules present in the lipid bilayer, just as the proteins are present in the lipid bilayer, right? It's all because of these hopanoids. The hopanoids in the bacterial membranes are drastically different from eukaryote, or you can say completely different, or you can say it's the specialty of the prokaryotic cells to have these hopanoids, right? We are not talking about the archaea as well. I don't think archaea has these as well. So this is the specialty of the bacterial membranes, like, right? I mean, the archaea might have it, but wait, let me search it. I should have searched it before. Does archaea have? I should have done that before. This is a good question. Yeah, just as I was saying, the archaea don't have the hopanoids, right? This is the specialty of the bacterial cell membrane. Now the question is, how are they different? Normally the hopanoids are amphoteric lipids. Not the amphoteric, they are totally non-polar. That's why they are present inside the bilayer and not up and down, right? It is present between them. So eukaryote and bacterial cell membrane have what common is the bacterial other membrane proteins or lipids or carbohydrates, things like that. But they are different because they have hopanoids. Now the eukaryotic cells have cholesterol, right? Cholesterol in them, sterols in them. The bacterial membranes don't have that sterol in them. They have, instead of sterol, the hopanoids. That's what they call it. That is the sterol-like molecule. Now how are they different? If you look at this diagram, this shows the cholesterol and this shows the hopanoid. General bacterial hopapentatrol. Now you will be saying, wow, they look completely different. how do you say that it makes the membranes different? The problem is the similarity in them is that a precursor for both of them is the same thing. Now you will be asking what a precursor is. Precursor is the molecule, is the compound from which a certain other molecule arises or is made, right? Like if you make a cake, there must be ingredients for that, right? So, or you can say, how do I clarify this to you? Like when you make a DNA, right? 5 carbon sugar. You need a 5 carbon sugar, a phosphate, and the nitrogenous base, right? Like these are the precursor. Even in that, the 5 carbon sugar has a certain precursor, which is called a different thing. Let me not make it complex. So the thing that the cholesterol is made from and the hopenoid is made from is the same thing. But when there are chemical reaction that occurs within them, it makes a completely different substance. So in this case, if you can see, there is this 5 carbon missing. It's kind of not missing, it is removed during the creation. It is made from, both of them are made from quinoline. Here is the diagram. Sorry, not quinoline. What am I talking about? Squalene. It is made from squalene. If you wait, let me show. I searched it. Precursor here. Yeah, see, the precursor molecule for both sterols and hopenoids are same, and that is squalene. Now, look at this diagram. If you look at these, oh my goodness. Wait. So these are different types of hopenoids. Wait, let me show you how hopenoid is made from squalene. Let's see if they show us something. Oh wow, this was the diagram. Where was I searching it? Yeah, so it is like this, and it folds itself in a cyclic form, and it makes things like that. Sometimes it folds itself in the 5 carbon, like here if you look at these 5 carbons, right? But in this case, if you can look at it, it's 5 carbon and the remaining chain is sticking out of it, right? So here the same case happened with them. If this made the 5 carbon, the squalene made the 5 cyclic carbon chain, right? In here it made a 4 carbon cyclic chain. Not 4 carbon, I mean 4 cyclic chain, right? And the remaining is sticking out. Now here it has the other remaining chain, but who can say if it is of squalene or if it is of something else? And if you look at them, they don't have that much hydrogen bonding, while this one has it, right? Well, in this case, the OH is attached to this side, of course, because it is a 4 cyclic carbon, right? Think about it. Now, one thing that is different was this. The bacterial membrane has sterols, while the eukaryotic one and the archaeal one don't have hopenoids, while the eukaryotic has sterols for that, right? Like for example, cholesterol. These compounds are made to stabilize the membrane, the functioning of membrane, because you know that the bacterial membrane is made from lipid bilayer, right? So it has the further more lipids to stabilize it even more, right? Now they are saying that these hopenoids are very important because they are very in high quantity. If you look at this number, 10 raised to power 11 to 12 tons. They are found in tons amount in the sediments under the soil, right? And the total number of carbon in all living organisms combined. Okay, we are talking about the organic carbon. If you combine all of them, that is 10 raised to power 12 tons. Almost or completely the same as the hopenoids. Think about it. Isn't it astonishing how the hopenoids, that are just the small portions of the cell membrane, are present that much abundant in the sediments, right? While the carbon that makes the whole body of the living organisms are exactly the same number as them. Now, of course, they are saying that it is important for ecologists and geologists because it helps in the formation of petroleum, right? So that's why it is important. Like, it is not something smaller to be say, oh yeah, bacterial cell membrane has hopenoids, what can it do? Now they are talking about that this cell membrane structure. If you look at this, you know how it is distributed, right, in the cell, like here, 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 here. It is not homogeneously distributed, it is not equally distributed. Like you say, right, there are three things with you. Wait, let me draw it. Excuse me. Right, you have A, B and C, and you are sure that you are going to put A, B, C in cell like this, right? Like this is your pattern. No, the cells don't have pattern for putting stuff like this. Like, if there must be some portion in the cell that have A more, and some portion that has B more, some portion that has C more. Like, you don't know which portion will have which quantity more, right? It is because of the environmental factors. If environment demands something in that area, so they produce more hopenoids in that kind of area more, right? And not just the hopenoids, the proteins as well, the oligosaccharides, or you can say things like that. They all are distributed randomly or concentrated in some area and in some not area. It is fluid model. Fluid. The fluidity is not just because of the floating of material present in the cell membrane. It is also due to the flexibility in having certain compounds in concentrated form in one area and some others, while when there is no need for them to be concentrated, they come back again because they move within that lipid bilayer, right? The third thing that is different in bacterial membrane is their foldings in the cell membrane. You would think that a whole cell membrane looks like this, right? A completely smooth sheet. But no, the cell membranes in bacteria have foldings. Like, you make, oh my gosh, I can't show you here, but like you fold it, right? Like, make foldings in it, put like dents in them, more and more and more and more and more, and then it covers the whole cell. Like, it will be better if I show you this picture. Look at this. Like, you see these grooves in them. Yeah, grooves might be the good word. Yeah, so these are the foldings of the cell membrane, and it is done to increase the surface area. Why is it done? And not in eukaryotic cells but in prokaryote. The reason is simple, because prokaryotic cells usually exist in singular form, work singular, right? Even if they are in chain form, they don't distribute their work. They do it solely on their own. So they need systems, strong systems, to do that single-handedly, right? Like, they need all the factories. And in prokaryotic cells, cell membrane is the strong organ in that aspect, because it has a lot of substances on the cell membrane surface, a lot of metabolic systems on the cell membrane surface. So the more the cell membrane is, the more the systems will, the more metabolic processes will occur. Right? Look at this. It also has, what is this cell? The first one was, which one? Wait. The first one is of the A is for the nitrifying and the photosynthetic bacteria. If you look at it, it's only on one form. I think this area is exposed to sunlight. Okay, so the first one is nitrocyst oceaneus with a parallel membrane traversing the whole cell. Parallel membrane? Oh yeah, you know now. You know. Parallel, like, if you look at from the above angle, you see the parallel lines, right? Parallel lines. That's why it's said to be parallel membrane. and the nucleoplasm with fibrillar structure. Nucleoplasm. Where's nucleus in it? Is this the nucleus? Sorry, what? Are they talking about this? Nuclear. Okay, we will come towards that later. This one is the ectothyroidspira mobilis, right? In extensive intracytoplasmic membrane system. Look at it. Certain areas have folding and some certain areas don't have. Now the other characteristic, the fourth characteristic, the bacterial membrane have that is different from the other eukaryotic or archaeal membranes are the mesosomes, and they are not found in all the bacteria. They are found in certain bacteria, not in all, and some certain bacteria, right? Now the mesosomes are these foldings, but they are folded in a more constricted area to make it more like vacuoles, and these vacuoles have different functioning, or you can say when there is a need for some kind of function, so they make mesosomes. Like it is not a permanent organelle in them, they make it. However, how much they need, they made it. Now the archaeal membranes. So guys, archaeal membranes have a very interesting structure, a very interesting bacterial membrane, and it will blow your mind. It will blow your mind how complex it is, even when it is not that complex, right? Now usually we see this structure. Look at this. If you think about a lipid bilayer, consider this is a phosphate head and these are the tails, right? Just as we studied. This is phosphate head and this is tail. Usually we have two tails with one phospholipid, right? With one head we have two tails. So this is one and this is the second, and this makes a double layer, right? One layer, double layer. But the interesting part of the archaea is that they make something very unique. They make single layers, monolayer. Look at this. Do you see them separate? Do you see them separately? Yes, we can see them. Do you see these separately? No, you can't see. This is a monolayer. This is monolayer. But the chains are so long that they don't need to make a double layer because they already have the phosphate attached to their end of the chain. Like it will be like this. If you take this phospholipid and leave it here and made another phosphate head at this side. So the heads will be hydrophobic and the heads will be hydrophilic while the center will be the hydrophobic region. The second important point in the archaeal membrane is they make branch chains. Do you see these branches coming out? Do you see these branches coming out? They are only in the archaeal membrane. Bacteria don't have the branch chain. Even if they have chains, they are not branched. Look at this. This is the difference between a bacterial or eukaryotic lipid membrane bilayer, right? So one phospholipid. Now if you look at this head, of course this will be the area, the glycerol which is attached to the phospholipid head, right? What am I saying? Phosphate head, right? So if you think about, there is a phosphate head at this side and the glycerol is attached to that area. I think for your ease I'll show you this here. Last stream I showed this. Wait, let me show you here. If you see the ball, this is the phosphate, while if you look at the chain, this is starting from the glycerol, right? Glycerol. Okay, what we were talking about is that, look at this, the chain is straight. The chain is straight, it doesn't have any branches coming out, but if you look at the archaeal membrane, archaeal phospholipid, it has branch chains. And if you go even more further, now there is an interesting, the one that I talked about, the monolayer, right? First, even before this, Tell me, why do they make branch chains while they don't? Do you see any differences in both the structures? Think about this. This is ester bond while this is ether bond. Ether and ester bond. What is the difference? Instead of me explaining to you, it will be better if you look at this. Look at this. Which one is ether? Which one is ester? Now this one is the ether. This one is the ester. Ester has the carbon double bond O. Double bond O is present in the ester, right? And the carbon is attached to another carbon. The carbon that has double bond O has attached to another oxygen, sorry. While the ether one has the oxygen and has C on both sides. C on both sides. I think it will be better for me to show you this. Look, this O is an important characteristic of the glycerol, right? This O is present in every area. Now, this O is connected to C on one side. Of course, even if this shows that CH2, it doesn't mean it is attached to H, it is attached to C. This O is attached to C as well. Now, if you look at this side, this O is attached to a C that has double O. But this O is attached to a C that doesn't have double O attached to it. I mean, of course, you know the basic structure, right? This doesn't mean that this area don't have. We know hydrocarbons has carbon backbone. So, of course, there is C, there is C, there is C, 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 right? All the way. So, this oxygen is attached to a C. Now, you understand the ester bonding and the ether bonding. So, which one do you think is more stable? The ester bond is stable or the ether bond stable? The ester bond is unstable because of this double bond O oxygen. It is more reactive, more chemically reactive, while the below one is quite stable because of its single bonding. We know 3 bonds are less stable than 2 bonds, and 2 bonds are less stable than a single bonding. We know this is the basic concept, right? Now, why did these don't have O's? Because these have branches coming out of that. It is all because of this ether bonding at the start. Now, let's forget about these, right? We know this basic structure. Now, if you go below, they are saying that the archaeal membranes that have phospholipid, these chains, there are usually about 20 carbons, right? Should I count? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Yeah, almost 20. I mean, it's, oh, okay, here, this one is as well. 17, 18, 19, 20. So one chain has 20 carbons, right? But the thing is, no, wait, carbon chains, 20 carbon chains. Is it carbon 20? I think. So, what was I saying? Usually have 20 carbons, right? And they usually have just 2 ether bonds. One here and one here. 2 ether bonds. But, there comes a but. And that is their ability to make, to attach to themselves another 2 ether bonds. Another 2 ether bonds at the other end, like at this end, but not directly. They attach to themselves the chain of another 2 ether bonds, right? This doesn't end here. Take another same or different, whatever, but the start should be same, right? Like the glycerol, and they attach themselves that. And this glycerol has a phosphate attached to it as well. That's why the pink area is phosphate head. So of course this area will have a phosphate. So if you look at it, it makes the tetraether bonds. If this one makes the diether bonds, 2 ether bonds, this one makes the 4 ether bonds, 1, 2, 3, and 4. Of course, forget about the OH. It's the same in the eukaryote and the archaea, right? Forget about this third one. Okay, this is not ether bond, by the way. This is hydroxyl group. So now if you look at this, 40 carbon chain is long. So archaeal membrane has the ability to make it even more complex. If they can make it complex by having branch chains, they can make it one step more complex by having the cyclic carbon chains in them as well. Now if the thing got complex very much, they fold themselves inside themselves, right, and make chains of them. Look, do you see? That's the pentacyclic. That means it has a cycle that has 5 carbons in them. It has all of the cycles having 5 carbon in them. Okay, why am I stopping? So anyway, this was just the general idea. Now, why do they do that? What's their problem? Why can't they remain this way? Why can't they remain single way? Why do they have to make it branch? Why do they have to attach another 2 ethers with them? And even after attaching 2 ether with them, why do they have to make cyclic chains in them? Why? What is the reason? The reason is stability. Environment is their biggest problem, and they survive that way because they make their outer covering more and more complex, more and more resistant to reactions, because these are chemicals. Of course, if they found some atom that is reactive to them, that is, wow, they are giving me electrons, yes, oh my god, I am taking the electrons. Now if they have a lot of carbons, they are saying, yes, I am giving electrons. So to prevent that reaction, they make complexes. They make their membrane complex, making, that the boiling point of the branch chain is higher than the straight chains, right? And it's because the bonding is simple in this, while the bonding in the branch is complex, right? It is preventing the inner carbons from reacting. So to make it cyclic, it prevents that even more. So if the bacterial membrane was different because of the inner molecules in them, the archaeal membrane is different because of the structure, because of how their bilayer is formed. So they do make bilayer, they do make monolayer, right? And even in monolayer, they make cycles in them and make it complex. And of course, they changes their polarity as well. So, yeah. So these monolayers are for the archaea that live in extreme hot environment. Now there are some archaea that do have monolayer that don't have these cycles, and that is for the moderate environments, right? Now there are also archaea that has some area with lipid bilayer, like this area, and some area with monolayer to make the complexity even more. So this was it for the general purpose, right? Now let me read it. Bacterial membranes are similar to eukaryotic membranes in that of their amphiteric lipids are phospholipids. They are similar to eukaryotic in what way? Because of the amphiteric lipids are because they have phospholipids in them. But they differ from eukaryotic membranes in lacking sterols. They don't have sterols. Instead, they have what? They have hopanoids that are sterol-like molecules. Now, the hopanoids are synthesized from the same precursor as steroids. And like the sterols in the eukaryotic membranes, they probably stabilize the membrane. Probably. Looks like they don't have an answer for that. That's why they say that they will probably do. More like they didn't do much experiments in them. Hopanoids are also interest of the ecologist and the geologist. It has been estimated that the total mass of hopanoids in the sediment is around 10 raised to power 11 to 12 tons. They are saying that it is important for ecologists and geologists because the mass of the hopanoids in the sediments is 10 raised to power 11 to 12 tons, which is about as total of the organic carbon in all living organisms, and there is evidence that the hopanoids that contribute significantly in the formation of petroleum. They are saying that they are the same in amount as the organic carbon, and they help in the formation of the petroleum. Now, the emerging picture of the bacterial plasma membrane is one of the highly organized and asymmetric system that is also flexible and dynamic. By asymmetric, they mean that they won't look the same. The half will not look the same as another half of, like the right side of the membrane won't look like the left side of the membrane, because the proteins and the lipids are floating in them. So of course they will have some concentration different in one area than the other, and that make it flexible and dynamic, different. So like if you look even in the same species, the bacteria, they will have a little bit difference in their cell membrane. Numerous studies have demonstrated that the lipids are not homogeneously distributed in the plasma membrane. Exactly what I was saying, they are not equally distributed. Rather, there are domains in which particular lipids are concentrated. Domains means there are areas where particular lipids are concentrated. It has also been demonstrated. Demonstrated means they have experimented on this, that the lipid composition of the bacterial membrane varies with the environmental temperature in such a way that the membrane remains fluid during the growth. Alright, so they are saying if you are growing with environmental temperature differences, so of course your plasma membrane will have different lipid composition, right? Now how? The bacteria growing at lower temperature will have fatty acid with lower melting points in their plasma membrane, right? Because they have lower melting points will mean they will not melt easily. You get it what I'm saying, right? Lower means in minus, lower minus. Back even more, like 10 raised to the power minus 6 is a lower melting point than the 10 raised to the power minus 46. So yeah, because they are trying themselves to resist to that lower temperature, they will make fatty lipids that will not melt at the temperature that they are living in, and so they will be resistant to melting than the lipids that are in normal temperature, right? Now if you look at the plasma membranes that live in the higher temperatures, their phospholipids will be the lipids that will not have low boiling point. They will have higher boiling point because they don't want to destroy their lipid structure because of the high temperature. So they will make it resist in a way that they will need even more higher temperature for them to boil. Anyway, so this is like, what I was saying. Although prokaryotes do not contain complex membrane organelles like mitochondria or chloroplasts, we know that they don't have these, but there is a replacement for everything, so even for them as well. So internal membrane structure can be observed in some bacteria. Again, it's in some bacteria, like cyanobacteria. These are the type of bacteria that do photosynthesis. Not all the bacteria do photosynthesis. So for photosynthesis, you usually need chloroplasts. But the bacteria don't have chloroplasts, so how do they do photosynthesis then? They do photosynthesis because there are membrane structure then. Now what was I talking about? Yeah, we were here. Plasma membrane enfoldings are common in many bacteria and can become extensive and complex in the photosynthetic bacteria such as the cyanobacteria and purple bacteria. Exactly, they are self-sufficient bacteria. They do everything on their own, so they need even more machinery. Look, the parasite don't need much of this stuff going on for them, right? Because they will steal it from others. I mean, bacteria take it from environment, or pathogenic bacteria take it directly, the nutrients from the humans, or you can say the host that they infect. But the bacteria that live in the environment, that survive in the environment on the material that is present in the environment and the complex materials, so they need to metabolize them in one way or another, and they need machinery for that. So that's why the example is the cyanobacteria and purple bacteria, because they have, or the bacteria that have high respiratory activity like nitrifying bacteria, because they process the nitrogen in them. Now these internal membrane structure may be aggregates of spherical vesicle or flattened vesicle or tubular membranes. Either the membrane is folded inside them, or either there are vesicles even in the plasma membrane. Guys, did you ever thought that there will be vesicles in the membrane, in the plasma membrane? Yes, there are. Either they are flattened vesicles or spherical vesicles, whatever they may be, but there is a machinery in it for them. Their function may be to provide a larger membrane surface for greater metabolic activity. Again, the larger area is, the larger the machinery will be fixed on those membranes or those areas. One membrane structure sometimes reported in a bacteria is mesosomes. Mesosomes appear to be invaginations of the plasma membrane in the shape of vesicles, tubules, or lamella. Invagination here means like the inward denting of the membrane, or specific, sacs-like things. Well, they have used the names like see vesicle shape, but they are attached. They are not detached. The problem is that because vesicles are usually detached, they are not. So there are invaginations, right? Wait, invagination? I have something in my mind I don't want to say. Let me search it. Invagination meaning. Yeah, inside out and folded back on itself to form a cavity or pouch. Guys, my mind is going to, well, let's not say it. Right, I'm biologist, so I don't have any shame, but let's forget it. Anyway, I did say I will go back to that, but I don't think there is any need for them. This is just the example of some bacteria that has these, membranous structures and stuff like that. Now, many biologists believe that they are the artifacts generated during the chemical fixation of the bacteria for electron microscopy. Artifacts. They are saying that this happens only when there is a chemical fixation in the bacteria for electron microscopy. Artifacts mean these are the artificial things, like it doesn't happen in the environment, because who will prepare bacteria for electron microscopy in the environment? Do they need that kind of protective layering? But when we put bacteria for the electron microscopy, we prepare the specimen by doing, certain methods. And they are saying maybe that mesosomes are not natural and they are made, artificially by humans. Like it's kind of error in the microscopy. So many biologists believe that because mesosomes are not seen in many bacteria, almost all bacteria. Let's talk about the archaeal membranes now. One of the most distinctive features of the archaea is the nature of their membrane lipids. They differ from both bacteria and eukarya in having branched-chain hydrocarbons attached to the glycerol by ether links rather than the fatty acids connected by the ester links. Understand that? They are different because they have branched-chain hydrocarbons. They have branch chains. And another thing is they are connected by the ether bond rather than the ester bond, and I have gone in that in detail. Sometimes two glycerol groups are linked to form an extremely long tetraether. Sometimes they are saying, two glycerol combine from one end to another and they make tetraether, 4 ether bonds. Usually the diether hydrocarbon chains are 20 carbons in length. Usually it can be longer or smaller, and the tetraether chains are usually 40 carbons because you double it, right? Cells can adjust the overall length of the tetraether by cyclizing the chain to form pentacyclic rings. Guys, by the adjusting means maybe the chain is way too long to form that lipid bilayer. Like they need some kind of consistency in their membrane, right? The membrane can't be more thick and more smaller. Like there is a certain limit at which the membrane might work better, and they need to maintain that limit. So they to overcome that length, that long length, they make it to make pentacyclic rings. Okay, so these are rings, guys. I was calling them the chains for so long. So I have already explained these. I don't have to go dive deeper in that. Now, phosphate, sulfur, and sugar containing groups can be attached wait, I can't see. Can be attached to the third carbons of the diether and tetraethers. Now they are saying if you have the sugar containing sulfur or phosphate containing groups, right? If you have groups, functional groups like this, they are attached to usually the third carbon of the diether, making them polar lipids because these are polars. How do I explain it to you? It's because look at the phosphate group. We do have a phosphate head at these chains, right? And that phosphate head makes it polar. Do you understand? Polar. Polar means hydrophilic. They love water. So if the sulfur will act the same way and the sugar will act the same way, and yeah, they are usually at the start. Like skip this one. They are usually at the start, and the functional groups are here on the third carbons. See, this is the third carbon, okay? Or maybe if they are talking about just the chain, then the third carbon will come somewhere here, but I think they are talking about this. And they are saying that this makes the chain polar. That means it will make one side hydrophilic. Like the outer sides are hydrophilic and the inward sides are hydrophobic, making it a casual plasma membrane, making them polar lipids. These predominates in the membrane, and 70 to 93% of the lipid membranes are polar. The remaining lipids are non-polar and usually derivatives of squalene. Should I explain what is this? 70 to 93% of the membrane lipids are polar. Means the more area of the membrane is out and reacts to water and likes water rather than the area that is in the middle, right? Oh my gosh, stop it. Despite these significant differences in the membrane lipids, the basic design of the archaeal membrane is similar to that of bacteria and eukaryotes. There are two hydrophilic surfaces and hydrophobic core. Exactly. One hydrophobic core means the inner area is just single, so that's why it's called core, while the outer sides are two, so the hydrophilic surfaces. When carbon-20 diethers are used, a regular bilayer membrane is formed. Yeah, a regular bilayer. When the carbon is 20, because it's not that long, so it needs another piece to make it a double layer, to make the length that is needed. When the membrane is constructed of C40 tetraethers, the monolayer membrane with much rigidity is formed, as might be expected. From their need for stability. Why? Because the membranes of the extreme thermophiles such as thermoplasma and sulfolobus, which grow best at the temperature over 85 degrees centigrade, are almost completely tetraether monolayers. Archaea that live in moderately hot environments have a mixed membrane containing some regions with the monolayers and some with the bilayer. I think I already explained these, so I don't have to explain it more, right? That the hotter the temperature is, the complex the archaeal membrane will be. And so, good. We are done with this, so goodbye. Bye-bye. Hope you studied something.